Hi, everyone. Thank you all so much for taking time out of your busy schedules to attend our OAI.edu live webinar series hosted by the OAI CORE Knowledgebase. I also want to thank today's speaker, Dr. Jamie Collins, who is the Associate Director of the Orthopedic and Arthritis Center for Outcomes Research at Brigham and Women's Hospital and Associate Professor of Orthopedic Surgery at Harvard Medical School. The title of today's talk is Working with Data from the FNIH Osteoarthritis Biomarkers Consortium, a nested case control study within the Osteoarthritis Initiative. And following this presentation, we should have a few minutes for Q&A. So if you have any questions, feel free to type them into the Q&A box at the bottom of your screen. And if you can't see it immediately, you might need to click the three dots to see more options. And so without further ado, I'll let Dr. Collins take over.
Great. Thank you so much, Julianne. And thanks to Jeff for inviting me to speak. And we're going to talk today about working with data from the FNIH OA Biomarkers Consortium. I wanted to start just by acknowledging the funding. Scientific and financial support for the FNIH OA Biomarkers Consortium were made possible by the organizations that you see on this slide. And just as a brief outline, I'll give an overview of the nested case control study, including the rationale and study design. I'll describe what data are available and how you access that data, and then just spend a few minutes on analytic considerations for working with these data. And I'd also just like to acknowledge the OA Biomarkers project team shown on this slide. These were all of the people involved in phase one, in particular, Virginia Krause and David Hunter, who are the PIs of this project. And here are some key references describing the rationale and the study design. In particular, that third reference has a lot of detail on the study design, which we'll go through briefly today. So in terms of the rationale for this study, despite the clinical and economic impact of OA, there are currently no pharmacologic therapies approved by regulatory agencies to prevent OA or stop disease progression. Improvements in clinical trial design are critically needed to overcome barriers to the development of disease-modifying treatments. And refinement and improvement of measures of joint structural change based on imaging and/or biochemical biomarkers are needed to identify individuals with NEOA that are likely to progress radiographically and symptomatically, and to overcome the limited responsiveness of existing imaging biomarkers. So the aims of the OA Biomarkers Consortium Phase 1 study is to establish the prognostic validity of several imaging and biochemical biomarkers for NEOA progression. So we measured NEOA progression sort of at a later stage at 36 to 60 months from baseline. And the study investigated whether biomarkers measured at baseline were prognostic of disease progression, with the idea that these could be used for prognostic enrichment of progressors in clinical trials. And it also investigated whether short-term change in biomarkers measured over 24 months were predictive of longer-term progression, with the idea that such biomarkers may be candidates to assess as potential surrogate endpoints in trials. So is there something that we can measure earlier than radiographic progression that's still predictive of a clinically relevant outcome? So the study design, it's a nested case control study within the OAI. And so by nested case control study, what we mean is that all participants were selected from the larger OAI study. So it's a case control study. So the case control status was based on disease progression. So that was measured with radiographic progression, which was a loss of medial minimum joint space width of 0.7 of a millimeter by 24, 36, or 48 months versus baseline. And this threshold was based on the OAI-specific smallest detectable change. Cases were also defined by pain progression, which was a persistent increase versus baseline in total WOMAC pain score above the minimally clinically important difference of 9 points at 24, 36, 48, and 60 months. And to meet this persistent threshold, that increase had to be maintained at least 2 time points. So the primary case definition was knees having both radiographic and pain progression. And so that's what we sort of deem our primary progression status in this study. And then the primary control definition was knees that did not reach criteria for both endpoints. So the controls included knees with X-ray progression, but not pain progression, X-ray only progressors, knees with pain progression, but not X-ray progression, so pain only progressors, and knees with neither X-ray nor pain progression, so a non-progressor. So again, these participants were selected from the larger OAI cohort. So out of the 4,796 participants in the OAI, excuse me, the first major inclusion criteria was that all required serum and urine specimens, X-ray joint space width, and MR imaging and WOMAC data at both baseline and 24 months. And so, just to start, this is quite a select cohort. Participants, in order to be considered to be included in this nested case control study, had to have complete biomarker data at both baseline and 24 months. Um, and then from there, eligible knees had KL grade between 1 and 3 at baseline. And from here, these knees were sorted into 4 mutually exclusive outcome groups. So again, those 4 outcome groups that we described on the prior slide: X-ray and pain progression, X-ray only progression, pain only progression, and neither X-ray nor pain progression. Knees were frequency matched by BMI and KL strata. And again, further details are available in that reference at the bottom. I think the main thing to point out here is that this is not a random selection. If you look, for example, at the X-ray and pain progression on the far left here, there were 252 knees that were potentially eligible based on X-ray and pain progression, and 194 of those were selected. And if you go all the way to the far right, there were 943 knees that were potentially eligible of non-progressors, and 200 of those were selected. So this nested case control study oversamples cases, and we'll come back to that when we talk about the analytic implications, but that's just something to keep in mind with the sampling strategy, as that our progressors or our cases are oversampled relative to the full OAI. So what data are available? Imaging biomarkers were assessed at baseline 12 and 24 months, which includes MRI, semi-quantitative assessment of MRI with the MOCS scores, and then quantitative assessment of cartilage, bone shape, subchondral bone area, and meniscal volume. And for X-ray, we have subchondral bone trabecular integrity. It's a lot of data and it can get a little bit confusing. These measures were assessed by several different groups, and each set of biomarkers is in its own data set. So for example, the semi-quantitative scores, the centrally performed longitudinal semi-quantitative readings with MOCS, that was performed by Boston Imaging Core Labs, and that's in one data set. Chondrometrics did measurements of cartilage volume, thickness, and other associated measurements, including subchondral bone area, and those are in its own data set. So when you go to download the data and work with the data, you just have to keep in mind that for each vendor or each group that did an assessment of these data, each has its own data set in the data download. And so if you're looking for something, you may have to look across several data sets to find what you're looking for. And you can see, again, just a description of what's available here: measures of bone shape, subchondral bone area, cartilage and meniscal volume, measurements of subchondral bone plate shape and curvature, etc. Biochemical biomarker data are available, again, at baseline 12 and 24 months. Data are available on 11 serum and 6 urine biomarkers, in addition to urine creatinine. And you can see the list on the right there. For the biochemical biomarker data, I would really urge people to take a look at the description. This is the document you can see in this bullet here. There's really important information on the assay methods, validation, and calculation of reference intervals. So there's quite a bit of documentation for this biospecimen data, and I would, again, urge you to look through that description in detail. And this also can get a little bit confusing because the data are available both for the FNIH cohort of 600, but also for 129 reference control samples of individuals without OA from the Johnson County Osteoarthritis Project. So again, if you go to download the biochemical biomarker data, you'll get data both for the FNIH and for Johnson County. And again, people can find that a little bit confusing. So I would, again, just urge you to look through that description first to make sure you understand all of the data sets and documentation that are available for the biochemical biomarkers. If you've not worked with OAI data before, it can be a little bit overwhelming at first, but I think it's a good thing because there's a lot of description of exactly what you're looking at. So you'll see several files for each data set. So for example, here, if you were interested in working in that chondrometrics data, the quantitative measures of cartilage, you would start by looking at this descript.pdf. This is the description of the data set that includes biomarker assessment and quantification. So exactly how the markers were assessed, how were they measured, and it provides references to publications describing that. The next two are data sets. It's a SAS data set, and then I think this is a transfer file. Both of these should be easily read into any sort of standard statistical software package. Obviously, the SAS data set can be read with SAS, it can be read with R, with Python, Stata, etc. So there's two versions of the data set. There's a contents file that has just the contents of the data set, so the variable names and labels, and that can be really helpful just to orient yourself to the data. And then finally, the stats includes descriptive statistics. And so this is always a nice check to make sure that you have the correct data and then it matches what's in the descriptive statistics file. And so again, this can get a little bit overwhelming, as I showed a few slides ago. There's a separate sort of piece of information for each of those vendors. So if you wanted to look at the iMorphics data, you would see a similar number of documents for that. So when you do go to download these files, you do get quite a bit of information. And so this is just to orient you into what you're seeing there. To access the data and documentation, it's at that NDA NIH website where you get the fuller Osteoarthritis Initiative data. If you click on the full download, it will bring you to a screen that shows all of the different data that you can get, and there's a separate button for the FNIH project. And so this, if you go to download this, it says biospecimens. It actually includes everything. So I included a screenshot of some of the things that you would download if you were to click on that biospecimen SAS. And again, it's all of the data for the entire FNIH, including the clinical data, reference intervals. This is showing the bone shape from iMorphics, et cetera. So you'll get dozens of files, but this would be everything you need for FNIH. And again, I realize it says biospecimens, but this is going to give you everything if you click on downloading that. So I think it's important just to quickly go through the clinical data. So this would be that clinical_FNIH SAS data set. These data are one line per participant and one knee per participant. So again, just going back to that flow chart, one knee per participant was selected for inclusion into the study. So this clinical data set will have 600 observations in it. I think the most important variables that you need from the clinical data set is that case control status. And so this is in two places. It's in this case variable where it's a numeric variable with the grouping seen here. And then there's also group type, which is just actually written out in text. And so that first participant is in group 2, which is the joint space loss only progressor. And that last participant is in group 4, which is a non-progressor, so did not progress on X-ray or pain. These data can be linked, as I said, to the larger OAI dataset, simply using the ID variable. So it's the same ID in both datasets. The top is that clinical data from the FNIH download, and the bottom is the all clinical data from the larger OAI. So for example, if you wanted to get comorbidities and WOMAC function and merge that in with the FNIH data, you could do that by ID. So it's really pretty straightforward. The one thing to keep in mind again is that, again, one knee per participant was selected for the FNIH case control study. And so if you are getting data from the all clinical data set, for example, you have to make sure that you're pulling the right variable. So that first participant, their right knee is included. You want to make sure that you're getting the right knee score for WOMAC disability. And the second participant, the left knee, is included in the FNIH study. And so you want to make sure you're pulling the data for the left knee. And I'm sure if you watched Jeff's webinar last month, he probably went through all of this, but the way that the side is included in the OAI dataset depends on which dataset you're looking at, whether you're looking at the clinical data, whether you're looking at imaging data. So just a note of caution to pay special attention to the side that's included. Again, that will be in that clinical FNIH dataset to make sure that you're pulling the right information. So I wanted to just spend a few minutes talking about analytic opportunities with this data set. So the first, obviously, is modeling that case control outcome. And so we have, again, our baseline biomarkers and then short-term change measured over baseline to 24 months, where we'd be predicting that case control status. And again, this is the most straightforward way to use the data to model the case control outcome. So again, to examine associations between baseline and or short-term change with joint space narrowing and pain case control status. I think, a word of caution here for the FNIH data, but really for any OAI analysis is that um, many people have used these data, and there have been lots of different analyses, both using the available FNIH biomarker data, also by groups who've reanalyzed the images and quantified new biomarkers. So I think, as with any OAI analysis, it's a really good idea to do a thorough literature review before you undertake any new analysis. So what are some opportunities that I see with this data that maybe have been a little bit understudied? I think mediation analysis would be a really interesting way to use these data. And this is an example of a paper that was recently published investigating whether synovitis mediates the association between bone marrow lesions and knee pain using data from the FNIH OA Biomarkers Consortium. And so the question with mediation analysis is that we may see an association between an exposure, so in this case, BML size score, an outcome, which in this case was WOMAC knee pain. And the question that we ask is whether there's a mediator um, through which some of that association is actually mediated. So in this case, the investigators asked whether some of the association between BML size score and WOMAC knee pain is actually mediated through synovitis score. And I think the FNIH offers a really unique opportunity to do some of these analyses because we have longitudinal biomarker data at baseline 12 and 24 months. So there's really, I think, a really interesting opportunity for longitudinal mediation analysis. I think there's also an opportunity to apply novel methods to assess composite biomarkers using machine learning or otherwise. Um, And so we have all of these data. We've done logistic regression, we've done random forest, but are there novel ways that we can try to investigate composite biomarkers? And I've included a couple papers on the slide here that I think would sort of offer interesting approaches. I'm really interested in generalized additive models, which is a flexible approach that allows for nonlinear associations between predictors and outcomes. And so I think there's still a lot here in terms of trying to investigate combinations of biomarkers that may predict the case control outcome. So just in terms of analytic considerations, the question I hear a lot is, can I undertake an analysis other than predicting case control status? So what if, for example, we wanted to look at a different outcome? We wanted to predict total knee replacement or functional decline instead of pain, or if we wanted to do maybe a cluster analysis for phenotyping. Can we kind of reuse these data for another question other than that case control outcome? And the answer is yes, but with an asterisk. So you can, and I think it is totally reasonable to do secondary analyses of a nested case control study using a different outcome or answering a different question. You just really have to keep in mind that this is a nested case control study and not a random sample from the OAI. And as I mentioned at the beginning, cases were oversampled. And so when undertaking a secondary analysis, we really have to be mindful of the selection bias and sort of what population this generalizes to. And there are statistical methods for this. So, any estimate of incidence or prevalence is going to be subject to selection bias. The recommendation is to use inclusion probability weighting to account for the outcome-dependent sampling. So you essentially reweight your estimates by the chance that that knee got selected for inclusion into the case control study. And then another thing to do would be to stratify the analysis by case status as a sort of sanity check. So let's say, for example, you were interested in whether baseline imaging biomarkers were associated with functional decline. You might ask whether the association between that baseline biomarker and functional decline is the same for cases as for controls. And if it's not, then you really do have to start to think more about that selection bias and what does that mean for the generalizability of your findings. And again, there are statistical methods to deal with this. I've included a paper here that includes some nice methods for doing this inclusion probability weighting. I think the analysis itself is relatively straightforward, but actually calculating those weights is not something that's available in the publicly available data. And so that's something that you would have to work through. And I did just want to provide a little bit more information on the FNIH OA Biomarkers Consortium. So on the FNIH website, you can get an overview of phase one of the Biomarkers Consortium project. I'll also point you to this slide deck that's on the OARC website. Way back at the beginning of the project in 2015, the investigators gave one of the pre-Congress workshops at the OARC meeting on the project, and those slides are available on the OARC website. Finally, I would like to just mention that this nested case control study was phase one of the OA biomarkers project. In phase two, which was the progress OA study, we attempted to externally validate the highest performing markers from phase one in data from the placebo arms of clinical trials. So the progress OA study is not part of the OAI. It includes data from the placebo arms of clinical trials. And again, we attempted to externally validate the highest performing markers. We are working on making those data publicly available and working with Jeff to make sure that once we do make those data publicly available, we can get the word out and let everybody access that. But in the meantime, I've highlighted a publication and the FNIH description of the Progress OA study. And then it's sort of a full circle moment today in talking about phase one of the Biomarkers Consortium. We're recording a podcast this afternoon with David Hunter for his Joint Action podcast that will describe the third phase of the Biomarkers project, in which we're attempting to get data from both the placebo and treatment arms of clinical trials to investigate some of these biomarkers. So be on the lookout for the announcement about the third phase of the trial. And again, that will go up in the coming weeks on David Hunter's Joint Action podcast. And so I wanted to end with the announcement for the next speaker in this series, Dr. Michelle Yao, who's an assistant scientist at Heber Senior Life, an assistant professor of medicine at Harvard Medical School, is going to speak next month about genetic data resources in the OAI, a primer. For those of you who don't know Michelle, she's really one of the preeminent researchers in OA and genetics, and I think this is going to be a really fantastic seminar. And so with that, I'd like to say thank you, and I'm happy to take any questions.
Well, thank you so much, Dr. Collins, for that excellent presentation. So we'll move on to Q&A. If anyone has any questions, feel free to type them in the Q&A box. I have a question. Can the FNIH MRI-based data be merged with other projects that measure the MRIs?
Yes, that's a great point, and something I probably should have put on the slides. So there are several other studies that have done MRI assessments in OAI. The one that comes to mind is POMA, which I can't de-abbreviate at the moment, but I think that was looking at people without OA at baseline and predicting long-term total knee replacement. And so those data can certainly be merged together. I think there is some overlap across the studies, though. So if you see, semi-quantitative imaging data from both POMA and FNIH, there is overlap in who was included in those. And so you want to make sure that you're checking to make sure you're not including somebody twice. But those data can all be merged. It's the same study ID for all of the data sets.
I'm not seeing any other questions, but at the moment. So I want to say thank you again so much for coming and giving our talk today. And also want to thank you all for joining and attending this live webinar. As Dr. Collins mentioned, next month we have another webinar with Michelle Yao. I sent a link in the chat to register. So definitely hope you can make that if you're available. And as I also mentioned earlier, we have a post-webinar survey that should populate after the end of the meeting, and we'd really appreciate if you take the time to do that. So without further ado, thank you so much, and we all hope you all have a great rest of your day.